Welcome, Joan Edwards.
Thank you for getting it sorted out. And thank you, Ron and Susan, for inviting me. It's really just a pleasure to be here. I'm going to talk about conserving our New England's amazing spring flora. And there's sort of three parts to my talk, so you can sort of follow along. And you can go to the next slide. Thank you. I'm going to start with some vignettes from our spectacular flora. Some of them are very, very common plants, some aren't even native, but I think the point of showing you these vignettes is to show you how if you look closely, virtually at almost any plant, you will find something absolutely amazing and astounding about it. Then I'm going to talk briefly about twin environmental crises of both climate change and biodiversity loss. We hear a lot about climate change, but we hear less about the importance of biodiversity in our lives and how critical it is really for all humanity. And then I'm going to talk about some ideas for conservation practices that my research has pointed to. So to start with, next, I'd like to show this picture of skunk cabbage drawn by Georgia O'Keeffe. Because I do a little exercise in one of my classes where I have the class go and look at this painting. We happen to have it in the Williams College Museum. And I have them write what they see about skunk cabbage. And they write lovely essays, and they talk about the color and the shapes and so forth. And then I take them into the field eventually during the course of the class, and we actually visit skunk cabbage, and I also lecture on it in class. So they know in detail the life history of this plant, and I have them write a second essay, saying, what did they see? And a lot of this is following what Georgia O'Keeffe herself really, the rule of thumb that she went by. Next. So this series of slides, this series of stamps came out, the U.S. Postal Service, and there's Georgia O'Keeffe with her giant poppy. She drew a series of large poppy flowers. And there's a quote from her, "Nobody sees a flower, really, it's so small, we haven't time." And to see takes time, like to have a friend takes time. Next. And so this was her sort of mantra. If I could paint the flower exactly as I see it, no one would see what I see because I would paint it small, like the flower is small. So I said to myself, I'll paint what I see, what the flower is to me, but I'll paint it big, and they will be surprised into taking the time to look at it. I will make even busy New Yorkers take time to see what I see of flowers. So, next. So my mantra is to look closely, and that's something everybody can do fairly easily. I always carry with me, although I don't have it on today, usually I do when I lecture, a loupe. This is my favorite one, Bausch & Lomb Coddington 10X hand lens. It's basically indestructible, and you can look very closely. And I just point this out because people misuse loupes all the time. They hold it at a distance rather than having it close to their eye and then bringing what they want to see up and looking at it closely. Next. So what I'm going to do is look closely at 5 different plants first and give you vignettes. 2 are very common plants. They're in bloom now, or maybe I haven't actually seen dandelions this year, but I'm pretty sure they're blooming now. Coltsfoot and dandelion, which are both very common plants. They're introduced, and a lot of people would like to get rid of them, but they're also really fascinating flowers. And then I'm going to cover various different native spring flora plants, the skunk cabbage, anemone or hepatica, which comes in blue and white, also comes in a pink form, and then 2 other plants, trout lily and spring beauty. These are all some of our earliest blooming native wildflowers. Next. So we're going to start with Tussilago farfa or coltsfoot, which you can find at roadsides and disturbed areas. They put their little yellow flowers up next. And you can see them, they flower before they leaf. So you see these flowering stems just coming out of the ground. Next. And the flowers themselves are, this is not one flower, but this is literally hundreds of flowers. That's the way aster family members are put together. And each individual little petal-like structure that comes out of the flower here is one flower. If you were to just pluck it like you would pluck the white part of a daisy, that would be one flower. And then in the middle, you see a different form of flower. So these on the outside are ray flowers, and in this species, they're all female. They're only female flowers. Those are the ones that will set seed. And in the middle are disc flowers, which you can see are 5 petaled, and they form a little vase. And they push up the pollen on the tips of the flowers. And these are male flowers in the middle. These little ones in the middle are buds that haven't opened yet next. And you can see this on the side too, really quite beautifully, the way the coltsfoot is put together. These are the female flowers. You can see their stigmas here, this sort of light yellow part here that they actually split into 2. Those are the stigmas. And these are the male flowers that have pushed up their pollen. They have plunger pollination in this family and pushed it up on the unopened stigmas that are non-functional in this case. And they put these little balls of pollen right on the tips of their flowers next. So the amazing thing about this plant is there's tremendous movement in the plant in order to increase their fitness, in order to make sure their seeds get carried, and also to annoy people that don't like these invasive plants invading their habitats. And they do something called circumnutation, where they do this circular dance. I'm going to show a video shortly. The flower heads, they flower low, so when they flower, they're just a few inches off the ground. Then when they're done flowering, the seed heads close up, and the whole flowering stalk tips over, like, so it's U-shaped from the side. And then it grows and grows and grows and grows, and eventually it straightens, and then all of a sudden opens into a globe of seeds. So we should see that on the video. I think if you click again, it should start the video. This was a student project. It's a little bit, okay, here they are. Here are the plants in the greenhouse now, and these are the flowering stems, so they flower first. And here you can see that circumnutation where they gyrate really around. You can see these are ones that have flowered. They've already started to set seed as they're developing their seeds. They're tipped over, and they just incredible movement. They're doing a dance, which is really quite incredible. And you can also see how rapidly they elongate. Whereas flowering is down here, we're going to see fruiting that takes place up here. And even in the dark, you can see they're elongating. They're still growing taller and taller and taller. It's really quite amazing that they do this. And if you were to just walk by them, you wouldn't see this. But you can see even they're elongating even in the dark. This is day 4. And they're still working their way up taller and taller. It's really quite phenomenal that they can grow that fast and eventually get their seeds up to where there's wind. They can be carried to new places. They're starting to straighten a little bit. Is it suspenseful a little bit? Oh, look, there's one here that is open. Now they open into that beautiful globe, like a dandelion globe. These are a little bit smaller. But you'll see these along gravelly riverbeds and waterways. But really phenomenal that these plants can do this next. Now we're going to move to the dandelion, which I think people are always amazed that dandelions are actually very interesting flowers. So these are dandelion plants. Everybody knows dandelion, but do you really know a dandelion? So if you look at it, you can see that there are over 200 flowers jammed into a dandelion flower head. And again, in this case, there are only ray flowers. They don't have those separate middle ones that the coltsfoot has. And each of these is one flower. Each of these little things would be plucked into one flower. And I had my students count some. It's well over 200 flowers in each dandelion head. And here you can see the stigmas, which are also quite beautiful. They split open when they're ready to receive pollen. And they form these little curlicue things in the center of the flower. Next. So if you look at a dandelion, this is the European dandelion, Taraxacum vulgare, and if you look at it under UV light, this is what it looks like. Next. It has a completely different image, and this is just to remind you that what you see in a flower is really different than what a pollinator sees. Pollinators, most bees and flies have UV vision, so they see a completely different set of wavelengths than we see. We don't see this amazing contrast between the center, and a bullseye pattern is often a common UV pattern. It's to say, okay, here I am, come get me. This is where you can get nectar. This is where you can get pollen. Next. And this is what a bee sees in a field of dandelions. And these are colorized. The bee might not actually see red and yellow. These were colorized. It might be black and white, but it's certainly bullseyes. And you see this amazing color pattern that's quite different than what we would see. And you have to sort of keep that in mind when you're thinking of who's going to visit the flower. What pattern does it have? What cues does it give to a pollinating insect? Next. So dandelions also do a dance, which is really quite phenomenal. And you just follow these notes here. The flower heads are low, and sometimes if you mow your lawn, the flower heads are really, really low. They stay below mower height. And then the developing seed heads actually hide in the grass. So if you have a flower head, it opens and closes every day, closes every night, and then when it goes to form seeds, the flower head curls up and it drops to the ground, and it sits next to the ground until it's ready to disperse its seeds. And when the seeds are ready to be dispersed, it straightens and then rapidly elongates up high so it can be dispersed by the wind. And that's why sometimes when you mow your lawn, you think you've gotten everything, and the next day you go out and all these dandelion seed heads that poke above the lawn. And I think the movie of this is next. Done by Sarah Clark, one of my students, and she put it to music. So you can watch them open and close. And sometimes we miss some watering, so sometimes they collapse because we didn't water them. Also do that circumnutation. Open during the day. I forgot to water. This is a student project. And there's one that came up here. And this one is cooking its seeds down here. Collapsed a little bit, but then it's going to come back up. They're not supposed to do that. This one's really wild. Look at it, it's just really amazing, their movements. And you don't think of them as being as dancing plants, but they are dancing. They're quite amazing. So I hope you'll think of dandelions in a different way. When I was at a conference once in Costa Rica, E.O. Wilson, who's the great biodiversity person and also was a champion of ants, was there, and there was a coffee break, and we went out into the courtyard and we were drinking little cups of Costa Rican coffee with cookies on the side. And E.O. Wilson took his cookie and he broke a piece off and he put it on the ground. And I said, well, you know, what are you doing? He said, always check to see what ants are around. So he was waiting to see the ants that came in. And then I asked him, I said, you know, a lot of people ask me, because I study insects and flowers, what to do if they have ants in their house. You know, so, you know, what would you recommend? He said, I know exactly what you should tell them. You should tell them that they should lean over and look carefully, and they will see an amazing organism. And I sort of say that about dandelions too. If you look carefully, lean over and look at them carefully, you'll see this really amazingly engineered flower with quite amazing contents. You can still weed them out of your lawn, but they're pretty amazing. Okay, next. Okay, now we're going to move to one of the iconic New England wildflowers, the very first native species to bloom in the spring. And we're going to look at skunk cabbage, Symplocarpus foedatus, which grows in wet areas, seepy areas, swampy areas around here. I'm sure you have plenty of them in your neighborhood. Next. And what's so amazing about them is that they produce heat. They produce the heat equivalent to that of a small mammal for a very brief period in their life. They'll heat up to 70 degrees. They're really quite amazing. And that allows them to bloom right even when it's snowing. They just power their way up. If you feel these spades that are ensheathing the flowers inside, they're really tough. They almost feel like plastic. They're really amazing. And they're this mottled purple and green, which supposedly makes them mimic rotten meat. And they have a very, very strong skunky odor. And they attract flies and bees and beetles to come into their flowers, which are hidden inside. Next. They can in part do this. They can heat up to such high temperatures because they have a pretty amazing underground root system. They have a rhizome, and then they also have these roots, and they store large quantities of starch, which they can burn really, really quickly in order to create the heat and produce heat. Next. So we actually put temperature monitors into the skunk cabbage, and I'm just going to show you a quick piece of data. So this is date over here, and this is temperature here. And we had 4 different probes, and the teal is ambient temperature. So this is the just regular temperature that you would get. And you can see for most days, the temperature at night at least goes below freezing. And the best one to compare this to in terms of a more spectacular skunk cabbage is the yellow skunk cabbage here. You can see almost always that the yellow line is above the teal line. And that's because this plant is producing enormous amounts of energy and heating up, creating a little island of heat, which could be a reward for a pollinator. It will also send out the odor into the environment to capture the attention of pollinators. But look at this differential at night. It's really quite amazing. This is almost 20-some, 22, 23 degrees here. And the flower is maintaining a temperature of 40-something. And some of the temperatures get as high as 70 degrees. It's really quite remarkable that it can do this. Next. The flowers are also equally fascinating. They're sort of like a pineapple, where the flowers are tiled onto this spadix in the middle of the flower. And the other thing that's interesting about these flowers, if you look at them closely, is that they're female first. So first they stick, these are the petals that are shaped like a box. And the stigma gets extruded up, and style and stigma gets extruded up, and often has actually a little droplet of pollinator fluid on it to capture pollen. And so the flowers do this first. They're female first, and then later they transition to male. So the stigma now is has done its job, and the stamens power up and poke up above the flower and dump quantities of pollen into the area. And this is a flower that has flowering head inflorescence that's completely in the female phase. And then you can see here it's shifting to male. The top flowers are already male, and the female flowers are about to transition to male. Okay, next. So what's interesting about skunk cabbage is it's one of the first, so it flowers first, it puts that flowering inflorescence up first, and then it puts up these beautiful, luscious green leaves that look like cabbage leaves, and hence the name skunk cabbage. But beware, because these leaves are armed with grooved rayfied crystals in specialized idioblast cells. Let me just explain what that is next. Is it not going forward? Yes, here we go. So if you have idioblasts are cells, so this outlines a cell here, and you can see all these things stacked up inside. These things that are stacked up inside are ratified crystals that are held in a mucilaginous soup. And they're double pointed, and some of them have barbs on them with reverse barbs. Some of them are triple pointed, but they're really basically micro weapons that the plant can use against herbivores. Next. So here's a close-up. This is again the idioblast cell. It's in a mucilaginous substance. So let's say a deer bites into it. The wall's going to break and water's going to go inside the idioblast, and that's going to create pressure. And the pressure then is going to cause these ratified crystals, these needle-like darts, to shoot out of the idioblast, and they'll go right into the mucosal membranes of anything that eats it. It's really a nasty thing to do, but it protects the plant. Next. So this is a movie. Let's see if you click again, maybe it will start. There we go. So some have already come out, but now look, do you see that? Isn't that just amazing that they can do this? It's incredible. And I had one year, when I was lecturing in the field on this, I had a class, and I said, skunk cabbage is edible, but I'm not sure how you get around the idioblasts. And unbeknownst to me, some students had collected the skunk cabbage, and they decided that cooking, of course, would get rid of the idioblasts. So they did a stir fry with the skunk cabbage, and they were really hungry, as students will be, and all of a sudden their entire throat was on fire. I mean, it was really, really bad. They're still alive now. They still joke about it. This was many years ago. But it really is a great deterrent for herbivores, except for one animal. Next. And that is our native black bears, it turns out, love skunk cabbage. And it turns out we think they eat it for a particular reason. Bears, of course, hibernate, and when they hibernate, their whole system just stops. And they build up these, what they call anal plugs, that are really full of hair and plant material and all sorts of stuff, and they have to really purge their system of it in the spring. And wildlife biologists think that bears use skunk cabbage as their ex-labs. It actually causes their system. I don't know how they deal with the ratified crystals, but they do. And you can go to YouTube and you can watch them eating this bears, which is really, really tanking up skunk cabbage, which is really pretty amazing. Okay, next. All right, so New England's earliest blooming native flower, skunk cabbage. It can bloom when it does and provide and photosynthesize when it does, because it has all these really special features that protect them against being eaten, that allow them to heat up and bloom really, really early in the spring. Next. And now, if you look at Georgia O'Keeffe's painting, I think you see a very different painting. Maybe, maybe not. I hope you do. But my students certainly did. They could see the heat emanating. They could see the sort of negative danger of the ratified crystals sort of exemplified in the very sort of dark image of this plant. Anyway, I think it's a really interesting thing. Your knowledge informs what you see in the world. Next slide. Okay, we're going to move now to Anemone aculoba, which is one of our spring ephemerals. It's not a true ephemeral because the leaves last year-round, but one of the things that anemone can do is it keeps its leaves. It has leaves that are winter green. They stay green all through the winter. Here they're starting to senesce and they're turning a darker purple, but they will keep photosynthesizing right through the snow. So another adaptation for our plants in the local flora. So last year's leaves persist through the winter, and enough light filters down through the snow that they can actually photosynthesize, albeit at a very low rate because it's cold, but they're also receiving some heat from the ground, so you can still have photosynthesis occurring. They're really quite amazing flowers. Next. Anemone aculoba here occurs in blue and white forms and also pink forms, so they're really colorful. I think they're actually blooming now. And has anyone seen them here locally? Yes, I think they're out now. Yeah, it's really, really cool. One of my absolute favorites. This is also a flower. We followed the pollinators for this flower, and they have the lowest diversity of pollinators, and I think it's because they bloom very early and they're aren't that many species out that are blooming. There's a small bee that visits, and there are bombolid flies. Those are the big hover flies with the long schnoz that come in and hover and visit anemone next. They also have a very distinctive UV pattern. So this is, again, the European one. This Bjorn Worslet has done the most spectacular UV photography of flowers. This is Hepatica nobilis, but you can see the contrast between the reproductive. Now this is particularly contrasty already, but you can see under UV it even shows up, it pops up even more. And in our species where we don't have this deeper blue, it would even show up even more in the UV next. They also, many of these flowers are very resilient for a day like today. So if Hepatica gets dumped on by the snow that we had today, no problem. They're easy to deal with. And this is a video. It's a bit long, but let's watch it. So this is a patch we were filming for visitors, and we didn't see any visitors, but we did see what happened overnight. This is 7 o'clock at night, 8 o'clock at night now. 7 in the morning, totally covered by snow. All the flowers bowed down and under this snow. And then gradually, as the snow receded and melted, these are the flowers. You can see their petioles that are bent down, and you think, oh, this flower's had it. But then you just watch. And this is where you really have hope, because they have this incredible resilience to deal with their habitat. There's so many challenges to blooming early and dealing with these very, very unpredictable weather patterns. And the flower, the petals are just starting to show here. Here's one showing. And there it pops up. And eventually, I don't show it all the way to the end, eventually they come right back to where they were in the first frame. They're really quite remarkable. And they bloom for a long time. Okay, next. Oh, keep going. This is repeated. Okay, we're going to look now at a group of flowers that are true ephemerals. These are flowers that you only see them for this brief period in the spring, and then they completely disappear. If you go back a month and a half later, there is no trace of them. And there's a whole group of flowers, including erythronium, which is the trout lily, and spring beauty, which is Claytonia caroliniana, that are only around in the spring and then they disappear. And if you think about it, there's a real challenge to living on the forest floor. You have only light coming in. They have to flower and do everything before the trees fill in and block the sunlight. And if you think of a sugar maple tree will block 99% of the incoming solar radiation. It's really hard to grow under that tree. But these little spring ephemerals, they push it as far as they can into the spring. They take advantage of that brief period when you have sunlight getting all the way to the forest floor, and they have a whole bunch of different adaptations for this next. So we'll look first at erythronium. Erythronium, very wisely, is a fair weather flower. It only opens in good weather. If there's bad weather, forget it, they just don't open. And they also open very late, like about 11 in the morning. There it is. And it closes early. By 5, it's done. There are visitors coming in. There are 3 visitors coming in that hit the flower, but I think it's hard to see unless we want to look at it again. And then by 5, it's closed up. Here's the repeating, so I think. Or it's just, yeah, 7 in the morning, 8 in the morning, 10 in the morning, 11 finally it opens. There's 1 hit, 2 hits, 3 hits. 3 different insects came in and visited it, and then that's it, closes up for the day. So it does that on a daily cycle, or it doesn't open at all if it's not good weather. Next. So if you look at erythronium, here they are in full bloom. This is a whole patch of them that we saw, and next. Here's the patch. This whole area is solid erythronium. And keep an eye, this is a good reference here, this bend in the river. And this is all erythronium, just solid erythronium. And that's 24 April. This is in Michigan next. And if you go back by 29 May, so a month later, they've all gone to seeds. You just see the capsules of the flowers, and that's it. And if you look at these seeds are really specifically designed to be carried from one location to the next by ants. Most of our spring ephemerals are ant dispersed, and that's really important to know for conservation purposes, because if you have an area that's isolated and doesn't have spring ephemerals, but they need to get there, nature's going to bring them in ants. And so, you know, you've got to think, how do we bridge that gap? How do we help? How do we assist new species coming in next? And then by July 31st, there's the bend in the river. It's completely shifted over to actually wild ginger is the common plant here that comes in after the erythronium, but there's no sign of the erythronium at all next. And this is just a little lesson on these ant-dispersed seeds. If you see fruits from our native spring flowers, you'll see that they often are two-parted. So this is the seed of Dutchman's breeches, which is a later blooming spring ephemeral. It's also a true ephemeral. It completely disappears by midsummer, or actually pretty early into the summer. This is the seed proper, and then it has this, in Dicentrus case, it has this very sort of feathery or many multi-branched eliosome, and these eliosomes are full of fat and nutrients for ants. So what ants do is they come in, they take this whole unit, they'll take the seed, they'll take the eliosome, they'll carry it back to their nests, and they will eat the eliosome and discard the seed because they can't chew through it. And that will essentially plant the seed near the nest of the ants next. That's true for all of our native trillium. All of our native trillium, this is stinking Benjamin, which take the time to lean down and smell it. It smells like a dog tongue. Really interesting though. And it attracts flies. It's really popular with the flies. But it also has this two-parted seed, where you have a seed proper, and then you have this eliosome, this fatty tissue, which really is nutritious for ants. And they'll take that whole unit and transport it to a new location. Next. And then spring beauties, which I also really love. These are flowers that really you need to look at closely. They're quite amazing. They have 5 sepals that you can't see here, 5 petals with often magenta nectar guides. So these are pointing to where the nectar is at the base of the petals. Often those lines that you see, you think, are they functional? Yes, they're functional. They're telling insects where to go to get the reward. They often have, although this is variable in populations, they have magenta anther sacs and often have pink pollen, which is also really, really pretty cool. If you look at an individual stem or inflorescence, the flowers develop sequentially on the inflorescence. Why would they do that? They don't want to put all their eggs in one basket. Maybe one of their flowers blooms today, there are no pollinators out, but their next flower blooms 2 days down the road, and there are lots of pollinators. So plants are really good at hedging their bets. And here you have the earliest blooming flower here. This will last 2 days, and it will be male the first day, female the second day, and then it will close up shop. This next one will bloom after that. That will be the next flower to bloom. And there will be a whole sequence of flowers that bloom over time in order to affect reproduction next. So looking closely at these flowers, they're really just stunningly beautiful flowers. These, I just am showing these 2 flowers in different stages of development. This is a 1-day flower that's in the male phase, and you can see it has anther sacs that are full of pollen. And most importantly, the stigma is closed up. It's a 3-parted stigma, but it's closed shut. So this is in the male phase. Day 2, these anthers fall further back. They typically have less pollen on them. Hopefully someone's collected them. And the stigma opens into this 3-parted stigma, and it's ready for the female phase, which is pretty remarkable. Next. So here is the flower of our deciduous forest. It blooms before the canopy trees leaf. Flowers only open in good weather also, so they open and close. New flowers develop every few days, so not putting all their eggs in one basket. It's protandrous, male first, female second. magenta nectar guides, has a heavenly perfume. It's also worth just smelling, and if you're in a big patch of them, you can smell it in the air. It's a wonderful, if I were going to pick a perfume to manufacture, I'd try and mimic Spring Beauty flower perfume. It produces nectar at the base of the flower, and it attracts bees and flies as couriers of their pollen. It's a really amazing little flower. Next. So I'm going to switch, so I just wanted to illustrate how when you look closely, and really you can almost take any flower and you can do the same thing that I've just done with these flowers. You can really see amazing features and think about how is it adapted to its particular habitat, how does it fit, what kind of niche does it fill. But I wanted to talk a little bit about our twin environmental crises and sort of relate looking closely and knowing your plants to saving your plants in some way. It's very clear, this is a paper that just came out in 2023, and I really liked it because it put our environmental crises in a really nice triangle where you have three factors that are important if you take humans into account here. One, of course, is climate change, and we know that's happening, and it's happening in a major way. One is biodiversity, and all of the services that biodiversity provides for us. And the crisis, of course, is that we're losing biodiversity globally. Nowhere on the Earth are we isolated from loss of species in our natural systems. And the third, of course, is how humans are affected either by climate change or biodiversity, or how humans also in turn affect climate change or influence biodiversity. So I liked this diagram. It's very dynamic. And it emphasizes both climate change and the ecological diversity and biodiversity of our communities is really important. We think of the kinds of things that biodiversity provides for us, oftentimes free of charge, and things that we're not actually paying for. Carbon storage, of course, is big. Think of all of the carbon that's in a giant maple tree or an oak tree. Think of how it provides clean water and flood control. New York City is the best example of that. New York City depends on water that's filtered in the Catskills and gets taken down to New York. It doesn't really even need treatment, and it's providing water for millions of people. It provides health and well-being. Even just looking at biodiversity makes you feel good, or makes me feel good. Climate control, a giant oak tree can cool as much air as 10 air conditioners. So, why not have an oak tree instead of 10 air conditioners? It creates soils, rich soils that also provide important ecological services. It, of course, is a source of foods and medicines for us. And the genetic diversity of biodiversity, the natural genetic diversity, is really important in terms of maintaining crops and so forth, and providing new crops. Next. If we look at a map of the United States. This was a graphic that was put out by the New York Times, and I really like it, but it shows where biodiversity is most at risk in America. And you think of some places where biodiversity is at risk. You don't think of New England as being a place where biodiversity is at risk, but it certainly is. It's significant areas in New England, so I just sort of blew up this portion of the map so you can see. And of course, Massachusetts is here right along the mountains, of course. Really, really key. Cape Cod, really, really key. So all of these areas are really important in terms of biodiversity loss. This includes animals and plants, but still, just to say it is here, it is in our neighborhood, and the fact that you have so many rare plant species, even in this area, I think is really important. You live in a really important part of Massachusetts. Next. We know from looking at studies, long-term studies of flora, that we are losing species. We don't think of them. You think of the rainforest as losing species, but you might not think about the species we're losing in New England and New York. This is a paper published in Rodora in 2019, where they really looked at historical records and tried to chart the changes in the flora. And a couple of highlights from the abstract. We find floras across the region have lost on average 1/4 of their native species. That's a local extinction. It doesn't mean the species is extinct, but every population you lose genetic diversity. And it's astounding to me that that's the average. It varies a lot, 3.5 species from the Finger Lakes region, so that's low, on the low end. But the New York, in Staten Island, New York, it's 53%. They've lost 53% of their native species. The other thing that's happening is we're having a grand mixing of floras globally because we've done so much trade, we've introduced plants, partly sometimes for because they think they will be good for us and then they turn out to be invasive and not so good for us. But the contemporary floras have a higher percentage of non-native species. The percent of non-natives has increased from 1.5% to almost 20% of our local flora. So this is also changing the biodiversity in our world. Next. Threats to biodiversity. This is another case where you have, here is a paper where they focused on climate change. This was a study where they compared the current vegetation in Thoreau's woods to what Thoreau saw and what they could show, and then they placed it on an evolutionary tree, which I thought was a really brilliant thing to do. So if you look at, this is a tree and it's in a circular format. So, but the important thing is in this tree, the things that the tips that are closest together are also more genetically related, they're evolutionarily related. And here you can see the aster family, and if they've declined, they're in red. So you need to focus on how many red tips do you have on this circle. And you can see lots of members of the aster family have declined, huge numbers. And they cluster by families. There's certain families that are more susceptible to climate change than others. And that sort of makes sense in terms, if you're genetically similar and you're affected by temperature changes or water changes, your closely related plants are also going to be affected. Next. And the cost of biodiversity loss is great. We all lose, and I'm sure I'm speaking to the people that already know this in part, but I just want to emphasize it. Biodiversity loss and its impact on humanity is just great. We all lose out if we lose biodiversity. Next. So I'm going to talk a little bit now about New England and particularly areas like Sheffield and like Williamstown. Williamstown, of course, I know more because it's in my backyard. And we have this 1830s map of Williamstown. And what you can see, the forested areas are in green. And what you can see is, in the 18th and 19th centuries, we basically deforested most of the East in a pretty major way. So what's happened through time is there's been a lot of reforestation in these areas. Next. So if you look at Williamstown today, there is much more. You could just compare along the Taconic Crest here. There's much more now that's preserved. The issue is, of course, it's secondary growth, not primary growth, but nevertheless, we are increasing forested areas. We have more protected areas than they had back in 1830. And one of the issues I look at in this system is, can we create corridors and reconnect fragments? Because fragmentation is really harmful to the loss of biodiversity. Next. And of course, this is Sheffield. I don't know it as well, but I was really impressed by how much green there is on this map. You guys are doing an amazing job, particularly down here. It's really, really cool. Isn't Bartholomew's Cove here? I'm not sure.
On the little to the, yeah, more over there.
Over here?
Down by where, I'm sorry, I feel right a little bit.
Over here?
Yeah.
Okay. Anyway, you have rare species there, and you also have a lot of green. But you still can continue to connect patches, no question. And you can also create new patches, which I'm sure you're working on, which is great. Next. And the last thing I want to talk to is considering communities, whole communities of species together. And one of my areas of specialty is studying pollination biology. And the question we're really asking here is, can we protect whole food webs? Because if you connect all the organisms that depend on each other. Of course, you can save both groups of organisms. And if we look at the flora in temperate regions, 87% of our flora is pollinated by insects and birds. Next. And we also have, and I've gotten into this, I started out as a botanist, but I am absolutely fascinated by the diversity of insects. I think they're harder for me to get into, but I've gotten into them in a big way. We have an amazing array of insect biodiversity, native insects. They also are given a short shrift in the world of biology. Flowers, showy flowers you can see and you can appreciate, but people look at a wasp, or they look at a tiny bee and they say, maybe don't think of it as being that important. But what I've learned is that flies, even this guy, this is a tachinid fly, a really hairy guy, with it's maybe a half an inch long, it's pretty big, or beautiful flower flies, syrphid flies, are quite amazing and also really critical for pollinating our native species. Beetles are also important, though they're voracious pollen eaters, and of course bumblebees and native solitary bees are really important. Next. So I have a couple of different projects that I've looked at. One is how do you manage a field to maximize floral resources for pollinators. I'm just going to talk about this briefly. This is, you can see here, this patch of unmowed area, and it's pretty late in the season by now, but it has a lot of goldenrods, and the diversity of flowers in this plot is really high. And then you can see in the background area, now this was a mode for hay in this year, now it's not mode for hay anymore. But you can see the diversity of flowers is much, much lower. And so one of the patterns we tested was to see if you wait to mow, can you cash in on the floral resources for pollinators next? So we did actually controlled plots, and these are plots that we waited to mow until after the first frost. These are plots that we mowed in late July. And you can just see from the dot pattern, and we scored, this is in the fall, we scored 4 different species of goldenrods, the grassleaf goldenrod and 3 other solidagos. And you can see clearly that if you wait to mow, if there's no reason to mow a field early, sometimes there are, if you're haying, there may be really good reasons to mow early. But if you have a field and you don't need to mow it early, why not wait? Why not wait until October and allow all of these flowers to bloom and be incredibly rich resources for insects at a critical life time for them, time in their lives, when they are trying to collect as much pollen as possible and make it through the winter. Next. The other thing to know is who's visiting the flowers. So this, in studying pollination biology, I really wanted to just be a flower and record everything that landed on my face. That would be the best. Because, if you've ever tried to watch insects visiting a flower, it's really hard, right? You come close to the flower to see what's visiting it and it flies away. So it's really, really hard. And I've developed this method where I use these little portable waterproof cameras that you can put out, and you can take a picture every 3 seconds, and you can get a time-lapse video of every insect that visits a flower, and you can do it for the entire bloom. This was really a dream come true in so many ways. So next. So here are some videos. This is bunchberry dogwood, which is an amazing plant. It's the world's fastest blooming flower. I should have put the video in of the flower. But you can see that the video allows you, if you were just looking at this, it's speeded up a little bit, but you would have a hard time, partly because there's more than one insect visiting, right? So it's going to be hard to actually follow all the insects that come in, and you certainly couldn't keep track of which flowers it visited. Next. But here you can see this one syrphid fly comes in at the blue, visits all of the flower inflorescences that are in green, and visits this red one and then exits off the screen. And if you have a video, you can keep track of the flowers it's visiting, you can keep track of the path it took, you can really get an enormous amount of data, and you can score the entire video, which we've been doing by hand, but I'm hoping AI is going to take over that job soon. I think it'd be a really good use of AI. And you can get whole pollination networks, and I'm going to show you just one for a local plant next. This is a network analysis for Viola canadensis, one of my favorite native violets. It's a later blooming violet that's purple on the back and white in the front. Has beautiful nectar guides that point to where the resources are for a pollinator. And these are just two patches in the same year. So this is one patch here, and this is another patch here. And then what I did is I arranged all of the insects that visited both patches down the middle. So they're in yellow. And then all the insects that only visited patch 2 are here. And all the insects that only visited patch 1 here. And these patches weren't very far apart. And this is a pattern that I've seen over and over. I can have a patch here and a patch maybe 100 meters away, maybe 300 meters away, but not very far. And they get different insect visitors. And so what this is telling me is that each patch has some unique visitors, and even though they share some, they still have unique visitors. And that means there's heterogeneity in the pollinator populations. And if you want to conserve both the flowers and their insect visitors, you better do as many patches as possible. Because each patch might have a separate group of insects visiting. Okay, that's my last point on this, and then I think I go to a summary next. So our New England flora, it certainly provides beauty, amazement, and is the basis for infinite discovery. I guarantee if you look at any flower, any plant, really any insect, you will see something amazing that it does, and you could make a new scientific discovery. Our New England flora is biodiverse and creates a community that stores carbon. I haven't talked about that in this so much, but clearly it stores carbon, and the higher the diversity you have in a habitat, the greater the carbon is stored. That's been shown over and over again. It filters air and water, and it maintains genetic diversity critical for persistence. Despite this, we're losing biodiversity. We need to conserve our flowers by saving every patch. We need to connect patches to ensure saving patches, and we need to make new patches. So, and I have to thank, a lot of this work couldn't have been done without, I have had dozens of students and colleagues that have worked with me on these projects, and it's been a lot of fun. And we've had funding from both Williams and the National Science Foundation. Thank you very much. I'm happy to take questions if people have any questions. Yes. Yes.
I actually have two,
Yeah.
probably 3 questions.
Okay.
I'll just not be that annoying. The first one is, skunk cabbage. I've been told that they can live up to 125 years in their community. Can you speak to that?
I don't know that for sure, but I would believe it. Yes.
I didn't hear the question.
Skunk cabbage, she's heard they can live 125 years.
Oh.
I don't know how they aged it. Do you know the source of that?
No, I don't know how they aged it. It was botanist.
Yes.
Shared that information with me. I don't know where
Yeah.
it was sourced from, and that would not be, so many of us have skunk cabbage around us. I don't know that it's each individual flower or the community itself, because they were speaking.
Well,
a lot
community
about-
itself certainly could, but
Yeah.
I wouldn't be surprised if individuals could too. I think a lot of flowers are much longer lived than we think, for sure. Something like Jack in the Pulpit, individual plants I know have been plucked in at least 25 years. So, it's really, really pretty amazing. Yeah.
My second question is about trap lily, and just to what you sort of said at the end about the need to preserve the patches where these are. Trap lily, like if I've been in the forest and I've seen not as extensive as the patch that you showed in the video, but a great amount of trap lily, is that an indication of perhaps the more tendency towards old growth constituents in that forest? It's more of an older forest, it's a maturing forest, and shouldn't those places be preserved? I just think about that.
Yeah, well, I think old growth forests for sure. We need to preserve, we hardly have any old growth forest in the East. I think maybe you can count it on 2 hands. true old growth pre-colonial forest. We have one stand at Williams College in our Hopkins Forest. It's really tiny though, and it's only preserved because it was on such a steep hill, they didn't want to farm it, so it's really quite amazing. Not necessarily for trout lily. I have seen trout lily in open fields. And if you think about it, actually, if it's ant dispersed, and if there's an ant mound, you're going to get all those seeds coming in there, and then you're going to get a lot of a cluster of it. Yeah. You had a third question.
In the image that you were showing of mow your fields late,
Yeah.
right? Like you can wait until October. So one of the things that I think about even past that is mowing them down and then not having places for these precious insects that are breeding so rapidly to be able to winter over. So I always think about, do we need to mow them at all? I mean, I know that's probably offensive to many
Yes.
people.
No, I think it is hard, and I've been sort of struggling with that because whenever you manage a field, someone wrote me a letter, they said they wanted to manage their fields for flowers, for insects, for bobolinks. You know, there's this whole list of things, and I said, oh, I think this sort of goes beyond my knowledge. I think it is hard. So not all overwintering insects are going to be in that field. I've also done an every other year mow. So that's something some people have done, or they've mowed in strips on alternate years. So there are other patterns. I haven't tested them, but I think there are ways that one could adjust it to include those, for sure. Important thing to think about. Thank you for bringing that up. Yeah.
Skunk, cabbage, and porcupine, do you have anything in common because of those quills?
Oh, that's very interesting. I don't think so. I think they're evolutionarily independently derived. Oh, do skunk, cabbage, and porcupines have anything in common? Because they both, shoot these, very sharp things. It's a lot. Well, they're thinking along the same lines. And evolution selected both of them to be. Yeah.
My other question, mowing our fields in the spring,
Yes.
they shouldn't be mowed too early, and cleaning out your beds, your flower beds and things.
Yes.
You shouldn't do that too early.
Yes. Leave it all.
Yeah.
Yeah. I think it's a good idea. Yes. Yeah. Yeah.
I get very confused by some of the things people say about mowing, and in particular, to do it early sometimes helps clear out the invasives because they have they are succession plants, so they often seed and then they come up before the natives. So if you get a good mow in early, then that will potentially weaken them so that the natives have a better chance of coming in. So you do want to do an earlier mow than
Yeah. I think.
like April, not May, or May, not June, or something.
Yeah, maybe if you want to just chop off the tops of the invasives before anything gets bloomed.
Yeah, because they're very good at, as
Yes.
pioneer plants and all that. And also what I hear is that they can change the pH of the soil,
They can-
especially
yeah.
garlic mustard. So
Yes.
if you let them persist, then they will make it doubly difficult for the
I think
natives.
it's hard with invasives. I haven't
They all have
come to a good solution. Garlic mustard, I think, is best pulled before they flower, if you can. It's pretty easy to pull them. It's things like multiflora rose and barberry and Japanese knotweed is horrible if you're in that kind of habitat. Bittersweet, Japanese bittersweet are really pretty.
There's somebody that wrote a good book about all these different protocols for the number of inches even for plants.
Yeah. So I'm not as much of an expert on those. We still have, even when we mow every year, we have invasives coming in. They've come into our plots.
Oh yeah, I just think it's a tricky thing to try to give advice about it until we have a better sense. And I also
Yeah.
think that there needs to be better cooperation between all of us who are interested in this and highway departments, because the highway departments are responsible for a lot of the invasive persistence, I think.
Yeah,
So-
and I think there is a lot of land along highways that really
Yes.
could be put to good use.
Me too.
And I think that would be really, really good. But still, in the absence, mowing in July at least is really knocking the flowers back. And letting them grow up till October is good, and you mow, and you still get the invasives then to some extent. But I'm not sure we can wait necessarily, that's the thing. The other thing is to maintain smaller patches that you can actually hand weed. I mean, I actually go out with a shovel and dig up my multiflora
Me
rows. Yeah.
The other thing is for the ephemerals, we collect seeds and then distribute them if the ants aren't traveling far enough, you know?
I think you could.
As long
you
as
could.
they,
Yeah.
yeah.
The problem with transplants is we're not very good necessarily at figuring out where plants can grow best. And I'm reminded of a project we did on Pine Cobble when we put in, this is Williams College, we put in a housing development. And it was in a site where the hairy honeysuckle grows, which is a state endangered plant. It only grows in maybe 4 places in all of Massachusetts. And what we settled on was to say, oh, we'll dig up any of the ones that will be in housing lots that we're going to build houses on, and we'll transplant them. Not one of our transplants lived. And I think it's because we're not really very good at it in many ways, you know.
Yeah.
Yeah, maybe if we did it now, maybe we'd do it better. This was many years ago, but yeah. But those are all really good points. Yes.
Have you ever discovered any successful ways to help those ephemeral populations? Like, is there anything in particular that's been successful that you've discovered?
I personally have not done it. You know, it'd be fun to do it, but I haven't, no. I have sometimes transplanted them into my yard and they've taken, like bloodroots. My bloodroot population is fabulous, yeah. So, yeah. But I don't have a formula for it.
Thank you.
Yeah.
Nasami Farm is a good place to go. Nasami Farm, it's
Oh, they're great.
the growing, place in Whately for native
Native
plant
plants.
trust.
What's it called again?
Nasami Farm.
Okay, thank you.
Yeah, and you can buy live plants that they have grown. And it's only natives there.
So it's the Nasami Farm and Wiggins, it's
Yeah.
Good little way.
Yeah.
Okay, hold on. Well, this is the day for phantom technical.
But it mostly worked. It was fun.
Yes, it was fun.
Yeah, and I can yell. I'll just yell. Yeah. Any more questions?
Right here.
Oh, yeah.
This is prompted by today. We have daffodils and these other little white flowers. I'm not a botanist, but, and they got snowed on.
Yes, and they're fine, right?
And was there any evidence that that they were up right yesterday, they get snowed on, do they kind of go back into the ground a little bit because they've been snowed
I
on?
don't think so.
Bend over.
Daffodils, they bend over, but they'll pop back up again. For the most part, they will. Daffodils, as does skunk cabbage, have contractile roots, which is pretty good. I mean, pretty interesting. So that when you plant a bulb, let's say you don't hit the spot right, you just kind of put it in the ground. It will grow roots out, and then it will self-adjust to the proper height by either extending its roots down and pulling itself lower, or extending its roots out and then growing up taller, and putting down new roots. Yeah, so they're pretty amazing. But most of these early blooming flowers put antifreeze into their cells so they don't get damaged by something like a late snow. Yes.
I have two, but one is sort of piggybacking on the one about cutting for the insects that are wintering over,
Yes.
I do leave mine up, but at this time of year, I'm antsy to start clearing in my perennial beds, and I don't know which ones are more likely to hold an apple.
Pollinator.
And I don't know if there are 50 ones or if there's a list of them.
I don't know, that's a good question.
I did read
Hollow
that
stem
maybe-
ones are really good, ones that have hollow stems.
I don't know if it would be okay to just So some of them are really, to cut them down, I don't know if anybody knows where the critter would be most likely to be boring in there.
I don't know, but this is a good question and I'm gonna research it.
I know you'll have the answer for it, I just couldn't right in the dark. It's just now I really want to get one of those blues.
Yes,
And you said it was a Bausch and Lomb, but I think,
Bausch and Lomb, yes.
but is there a
Coddington 10X.
Coddington?
Coddington, Codding. TON.
Paddington?
10X, 10X magnification.
Mother's Day is coming.
Bausch and Lomb, yes, it's a perfect Mother's Day gift.
Thank you.
Yes, yes.
Actually, I just wanted to answer your question a little bit because I just watched with part of a class sponsored by the Native Plant Trust, and I've watched Xerxes videos and so forth.
Oh, yeah.
But the last one I watched, they were talking about, cutting it back, how far, and they suggest that you leave about the first 12 inches.
All right.
Okay, and this was interesting. They said if it has a flower on it now, it shouldn't have any pollinators in it this year. The point is to cut it to about a foot, say it can vary, and so that next fall, that's when the insect is going to use the hollow stem. That they're not going to bore into it themselves this year, so that if it has a flower on it, you could actually, you want seed heads for other reasons. But otherwise, you shouldn't be harming anyone who's overwintering. They want that stem for next fall, and that's when they're going to use it. So it's interesting that you don't have to worry about from that standpoint.
Yes.
With this conversation about helping the insects that everyone's talking about, I understand it's probably to try to get as naturally as possible, but I wonder how effective are insects, insect houses, like the little houses people make where they have little holes.
Yeah, I think you can put out bee nests. I think it's a great thing to do.
Yes.
Yeah. I don't know, I haven't charted to see how effective they are, but I've seen them many places and I think it's a good idea. So any hollow tube straws would really.
Actually, they're saying that when you have too many insects gathered in one of those hotel type things and it's supposed to collect a variety, it's very bad because diseases and stuff happen, and if you get mildew or whatever, and so it promotes
So they're small, spread them out.
tiny ones, yeah.
Any more